Hello, Assalamualaikum warahmatullahi wabarakatuh. I am Abu Hurairah. My metric number is 191900. Today I will present on the silver nanoparticles as an alternative of my antimicrobial product. First of all, nanoparticles are nanoscale materials that are built with unique physical and chemical characteristics and also properties. The electronic, optical, and chemical properties of nanoparticles behave very differently from those of each component by their larger scales. Nanoparticles of silvers have the range size of 1 nanometer to 100 nanometer. It is composed of a large size of silver oxide, which is Ag2O compound. It is commonly found in spherical, diamond, octagonal, and thin sheets shape. Silver nanoparticles have a wide range of applications, especially in biomedical sectors where it is used as a coatings for biomedical equipment such as keyboards and wound dressing due to their properties that emits low-level silver ion, which provides protection from bacteria since it has antimicrobial properties. To identify the silver nanoparticles, one of the methods is to the size and shape often measured using typically analytical methods such as transmission electron microscopy (TEM) and scanning electron microscopy (SEM). The typical state of silver nanoparticles can be obtained by analyzing the spectral properties of the solution. Careful monitoring of the UV visible spectrum of the silver nanoparticles with time is the sensitive technique used in determining if any nanoparticles aggregation has occurred. If the nanoparticles inside the solution have not agglomerated and have a spectral shape that is identical to the as-received suspension, the UV or visible extinction spectra can be used to quantify the nanoparticle concentration in the solution. The concentration of silver nanoparticles solution is calculated using the Baeyer-Lambert law, which correlates the optical density, or D, a measurement of the amount of light transmitted through a solution, with concentration. Due to the linear relationship between OD and concentration, these values can be used to quantify the concentration of nanoparticles in solutions. From this left graph, it shows the extinction, which the scattering plus absorption spectra of silver nanoparticles with diameters ranging from 10 until 100 nanometer at mass concentration of 0.02 milligram over milliliter. In the right graph, it shows the extinction spectra of silver nanoparticles after the addition of a destabilizing salt solution. With this, we can identify the amount of nanoparticles present in a solution. The toxicological study of silver nanoparticles. The growing of interest in the understanding of the relation between the physical and chemical properties of silver nanoparticles and the antimicrobial properties, and also its potential risks toward the environment and consumers. Due to the increased application of silver nanoparticles in consumer products, there is a large international effort underway to verify the safety and mechanism of the reaction of antimicrobial effects of silver nanoparticles. The ion release rate is a function of the nanoparticle size, which the smaller particles have a faster release rate. The higher temperatures accelerate dissolution and exposure to oxygen, sulfur, and light. In all studies to date, silver nanoparticles toxicity is much less than the equivalent mass loading of silver salts, which can be very beneficial since it may reduce the potential of causing a side effect when used in biomedical sector. Silver nanoparticles we focus on silver nanoparticles as anti-cancer agent. Synthesis through biological approach has been the most cost-effective, environmental friendly, and can be easily scaled up for large-scale synthesis. This is the synthesis of silver nanoparticles in biological approach. Biosynthesis of silver nanoparticles can be done through several methods by using either bacteria, fungus, and also plant extract to synthesize the nanoparticles. Among all of that, plant extract method is the best due to the simplicity in conducting the synthesis procedure, as it does not require the elaborate process of tissue culture, which reduce the probability of the culture to be um, infected by any outside agent. Two types of silver nanoparticles that was studied is AGNPS EPS AER and AGNPS EPS ANAER. It is tested for two cancer cells that were tested, which is breast cancer cell SKBR3 and also human colon cancer cell. The material used is the sample is from Melia dubia, which is a type of plant from Meliaceae family. It is used since the leaves of this plant contain alkaloids, carbohydrates, glycosides, phenolic compounds, tannins, gums, and mucilages. Medical plant extract has generally contributed the most in the drug discoveries, such as herbs and also others. It is investigated to synthesize the silver nanoparticles by leaf extract of the Melia dubia plants and tested of this effect of silver nanoparticles on in vitro fertilization anti-cancer activity against human breast cancer, SKBR3, and colon cancer cells. The procedure to prepare for this material is first, the cells of the plant will be plated in 0.1 milliliter of medium well on 96 well plates. Then it is go under incubation for 72 hours for cells to reach confluence. After this, it is continued to incubate in the presence of various concentrations of samples, 0.1% dimethyl sulfoxide DMSO for another 48 hours at 37 degrees Celsius. Once the removal of sample solutions and washing with phosphate buffered saline at pH 7.4, 20 milliliter, well 5 milligram over a milliliter of 0.5%, 3,4,5 dimethyl 2-thiazolyl 2, 5-diphenyl tetrazolium bromide cells, which is MTT. Phosphate buffered saline solution will added into the solution and medium. After 4 hours of incubation, 1 milliliter of DMSO will dimethyl sulfoxide will be added. Viable cells were determined by the absorbance at 540 nanometer. Measurements were performed and the concentration required for a 50% inhibition of viability was determined graphically. From the result of the experiment, it is tested onto human breast cancer cell lines and 3 human colon cancer cell lines. The cancer cells are totally proliferated, inhibited once tested. This proves that silver nanoparticles have high cytotoxin properties. The mechanism of AGNPS, which is silver nanoparticles, was reported such that the silver nanoparticles provide a perfect surface outside of the mitochondria for the univalent reduction of oxygen to superoxide from electron through the electron transport chain. Silver nanoparticles also has other mechanism where they bind to the proteins and DNA of the cancer cells. Reactive oxygen species and oxidative stress occur as an early event leading to NP-induced toxicity for the cancer cells, which can lead to death. This can lead to death of cancer cells due to the effects of ions released to the mitochondria, which inhibits the production of proteins for the cells, and nuclei, where they interact with the DNA of the cancer cells and eventually kill the cells. This graph shows the summary of the results where both of the silver nanoparticles attached to the mitochondria and the nuclei of the cancer cells. In conclusion, with the application of silver nanoparticles as an alternative for anti-cancer product, a new path can be created where the cancer cells can be contained and hopefully cease to exist due to the mechanism of silver nanoparticles, which act like a cytotoxic for the cancer cells. Hopefully, this technology can cure the cancers that we as a human being face right now. This is the reference that I have used for this project. That's all from me. Thank you.